So our first speaker is Dr. Riccardo Velasco from the Edmund Mach Foundation in Italy. He is a plant breeder, so he leads the molecular breeding program for apple, grapevine, and soft fruit, as well as the high throughput genotyping platform. He's also a leader of one of the EU projects and a national program on quality and nutrition in apples. He has many, many years of experience in plant genomics and crop breeding, and has worked on a lot of different, fruits and cereals and crops in his life. He's refereed over 100 articles and book chapters on genetics, genomics, molecular biology, and molecular breeding, and has also served on the board of several. He's been on editorial boards of several magazines. So there we are. Thank you very much.
Yep. Thanks for inviting me to give a chance to present some of our work. And actually, I would like to tell you a story of our activity in the last few years. Actually, we have been involved quite a lot in the fruit genomics. And we have actually found some of the product that we will have to speak about at the end of a talk. Very useful for our purposes. Actually, our institute is a research institute aiming among the goals to the production of new varieties in fruit crops, mainly apple, but also grape, soft fruits in general, like strawberry, raspberry, blueberry, and so on. And the department where I'm working with is the one that indicated in red. Do you have any pointer? Yeah. Great. Okay, this one indicated in red. So we are a department where, beside quite a few basic science and blue research, we are also organizing technological platforms which aim to breeding of rosacea, as I said before, or grapevine, but as well sequencing and genotyping, and that is the work that mainly performed in the last few years, and also plant transformation and the production of GMOs. At the moment, only for research, but actually I have nothing against to produce them also for production. One of the main goals, as I said before, is the genomic biology of fruit crops, mainly a production of new varieties of apple, grape, and soft fruits. We have quite a large breeding program. Every year we bring at least 8,000 new genotypes in the greenhouse. Actually, starting from a very large number, we would like to reach this number of plants brought to the fields, starting from, why not, 10 times higher, selected by molecular markers. The same is for grape, and most recently we have begun also with soft fruits. So how did we perform it to improve our molecular breeding program? Actually, we have been involved in 5 genome sequencing projects, started from grape in 2006, and the last 2 arrived at pear and raspberry. The pear has been published on PLOS ONE very recently. Raspberry is still ongoing, led by one Utah University. As I said before, from DNA sequence and genome discovery, we aim to phenotype our populations or our germplasm to identify, actually I have not yet finished, but thanks. I'm pleased about it. We identify genes that withstand to QTLs, which from time to time we are lucky to have exactly the gene function corresponding, and we can develop very fine tuning molecular markers, obviously to exploit the biodiversity. So I brought one case studio, which is the apple genome. Actually, don't forget all the numbers, but as a heterozygote genome, actually we have had quite a lot of difficulties to assemble it. And still, although we are already at the version 3, we have quite a lot of scaffolding, but we prefer to maintain them rather than join the wrong one with the wrong one, to maintain them and to link as many as possible of these contacts. As you can see, is over 75% of them have been correctly anchored to the 17 linkage groups or chromosome of apple through almost 20,000 markers. Clearly, working with these numbers is possible only because we have done quite a high throughput technological work. The genome, as I said, is pretty fragmented with respect to the usual homozygote genome that have been sequenced from many other crops, but actually is quite correctly anchored. And we improve it very recently, although one of the troubles we have faced is the fact that the apple genome is also, although it behaves very disomic in the subdivision of the chromosomes and inheritance, we discovered already after the early time we began to sequence the genome that actually the apple is a false tetraploid, actually, because although it was extremely useful with the work with the other rosaceae group like peach and strawberry to understand which were the ancestral genome of the rosaceae based on 9 chromosomes, and which were the derived then chromosome and duplicated chromosome in apple, because obviously apple has roughly double number of chromosomes of most of other rosaceae. We discovered that the chromosomes were pairwise, like the 13 and the 16, almost totally synthetic. So as you can see, is like to work with the tetraploid in assembly the genome. So it double the difficulty is not only the heterozygosity, but also the fact that we have to face with a very high similarity between different chromosomes. So how did we improve it? We had the luck that some time ago a good breeder in Angers has produced, Professor Spinaz produced a double haploid of Golden Delicious, the variety that we have sequenced, and supplies to us 2 of these double haploids. They are 2 different ones, and they have chimeras of the original Golden Delicious genome. We have sequenced with a depth of over 50x with Illumina, and we have the luck to assemble it and improve actually to up to identification mostly of the case of the 2 haplotypes present in the Golden Delicious. So these are the 2 double haploid, and these are the chromosomes that has been, so these are the 2 chromosomes, sorry, of the Golden Delicious. These are the 2 double haploids sequences. And we could identify the haplotypes, either blue or red, which correspond to one or the other haplotype. So also we have developed some software to have a very quick scoring pipeline, and produce the highest density physical, so genetic map with where the markers here represent the physical coverage of at the end of a converging map of 23 crosses with over 17,000 markers. So it's pretty dense, and it allows us to anchor those 65,000 contigs quite correctly, and to correct some of the mistakes that we were facing with when we did the version 1. As you can see here, there is chromosome 1. These are 2 parentals, Yanagold and I don't remember which other variety is this one. Anyway, these are the chromosome 1 of Yanagold and the other variety. And as you can see, there is the wrong attribution to chromosome 6, chromosome 15 or 16 of some, through some markers which were positioned wrongly, or better, markers were contigs which were positioned wrongly, and we could correct through the 17,000 markers, bring them back to the correct position on the chromosome 1. So we could align correctly along the 17 chromosome over almost 80,000 of the 80% of these contigs. So one, or better, 2 tools which were proudly produced in these last couple of years in the project Segregating, where we are leading the VP6, which is aiming to produce a chip, DNA chip. We produced together with Illumina a 20,000 SNP chip that you can run on Illumina iScan. And very recently, so it's not yet commercially available because the project, the European project not yet finished, is finishing in August, but after the summer will be also available. And you can see probably in at the booth of Affymetrix already this logo where the apple is also listed. So, the results of 480, so almost 500,000 of SNP chip has been done by sequencing by row, initially 14 varieties of the collection. So these spots are representing the 1,200 varieties are present in collections in our institute as well in other institutions around Europe. And we have selected some of them representing as large as possible variabilities. And then to go to the actually 480 kilo SNP array, we add another 48, so covering almost the entire representation of European apple germplasm. So by these two arrays, we have, with the 20K produced in 23 maps that has been merged in the slide that I showed you before, and with the 480K, we will characterize and propose some genome-wide association studies on the European apple germplasm. And this will be led by Marco Bink in Holland. So this is the processes that we have used. So with almost 10 million of SNPs, leaded to the 500,000 SNP that has been positioned on Affymetrix. So then to the real aspect, which is interesting for the, let's say, the more molecular biology and more molecular genetic aspect. So as a breeding institute, we are interested to breed new varieties, spatially resistant genotypes to the major diseases like apple scab, like apple proliferation, like fire blight, as well as some other quality traits, especially important for or some curious trait like the columnar one, based on like the self-thinning or other quality traits of a fruit. So starting from such kind of pretty dense and good referiment points with a very high density markers, we can reach and we can close up, be very, very close to the genes, if not inside the gene, like VF, which was actually presented here the first time in San Diego in the year 2001. We have been able to identify or be very, very close to most of the other 10 regions. Sorry, because it's in Italian, this one didn't realize it. I realize that right now. But there are, at least, believe me, these are 10 resistant genes in apple scab. So I promise, this is the only one in Italian. That is, again, in English. So they have been positioned, and thanks to the very dense map, as I showed you before, we have very, very close markers. And in 3 cases, like VR2 has been recently cloned in Switzerland, but VG, VB, and VM has been identified as one gene in 170K space, kilobases space. So most likely is the one that is responsible for the VG resistant. VB has been now identified a 300K, 300 bases space in the linkage group 12, and the VM actually is also close to be identified because we have identified just 3 genes in a space of 180 kilobases in linkage group 17. So having this very high dense map, we can reduce on the back libraries in a single back, even within the back, very, very close to the gene. If not, it's enough, obviously, a very, very tight, very close molecular markers for our purpose. The purpose is clearly to speed up the breeding, instead to wait something like 30 years before to have, like in the case of VF, the first resistant genotype, speed up by breeding by design some healthy plants which contain resistant genes. This is done by creating elite lines with the concept of Peleman published in 2003 in Breeding by Design. And as you can see clearly, we need to identify the genes that control the traits by linkage association through a screen of germplasm collection and support then the breeding with the mass. To do that, as you can see, we have hundreds, but most of the time thousands and thousands of plants to screen. And actually, with the aim clearly, which is still relatively skeptical in the apple field, to obtain a very good apple, very tasty, crunchy, sweet, juicy apple, and also resistant to several diseases, possibly with more than one gene, which help the plant to defend himself against also the same, like in apple scab, more than one gene which help the plant to defend itself. So we have something like 18 hectares in San Michele, and as I said before, we aim every year to bring in the field 5,000 to 8,000 genotypes. 5,000 to 8,000 genotypes to be brought in the field means that every year, the year before, you cross at least 25, 30, up to 50 different crosses. You produce something like 15 to 30,000 seeds that you have in a very, very short time bring from the greenhouses where you screen. Here there is no picture of the greenhouse, there is only a screen house, but in the greenhouse where you screen, maybe next slide, yeah, here, where you put to grow your seedlings, these seedlings cannot resist in this condition more than 3 weeks. 5 weeks in a very lucky case, but then either they die or they stop their growth, and that is a tragedy for the month after, of the year after. So what we have to do is to do something very quick, something very efficient, to screen, to help the breeders from the 20 to 30,000 seedlings that they have, how to select the ones to be brought to the field, which will be obviously not, cannot be too many, because we don't have more than 1 hectare to replace every year. So in this case, we have found very useful the Kappa 3G plant PCR and DNA extraction, both cases, because actually we have done the extraction in very few days by collecting. So the most time consuming was collecting just a very small, tiny piece of leaves, and we have screened. For instance, this spring, something like 6,400 plants within 3 weeks, and we were able to say to the breeder which plant from these trays, which were by chance 96 well trays, because they in this way correspond to a 96 well plate for the screening. So you do not have to record to name or number every plant, but it's enough to have a barcode to every tray of a plant, so they correspond to every 96 well plate. And we have screened for several, especially for microsatellites, because actually it works very well with microsatellites, the extraction, very clean. Several microsatellites which are in the near region of 4 resistances, gene responsible for the resistance to apple scab, 1 to downy mildew, and 3 for quality traits, which are crunchiness and storability. So, as I said before, we have found very useful both the extraction kit as well the plant PCR kit, and they allow us to reduce the cost to 42%, which is extremely important, because even if the cost is not extremely high with respect to the sequencing of a genome or whatever you do in a very high blue sky, high-level blue sky research, still having, for a breeder, having 10,000 or 20,000 euro or dollars cost every year is something, is important to reduce as much as possible. And clearly doing it in a 96 tray and 96 well plate, we were able to do in a row the results with a very, very low number of individuals that has to be repeated at once. So below 10%. Yeah, I'm finished. Actually, I brought here just a couple of examples of our products. These are plants that are not treated at all. So these are, these plants carry specifically one resistant to apple scab and one resistant to oidium. And this is a daughter, this is a sister plant of this one. As you can see, this plant is, again, not treated, but is absolutely devastated by apple scab. While his sister, they both belong to the same cross, coming from the same parents, is absolutely resistant. And these are the first results of triple resistance, pyramidized. In this case, oidium, scab, and fire blight. So actually, these are, I guess, the last slides, and there are thousands of people that I have to thanks, because as you can imagine, from the beginning of a sequencing project to the field work, there are very, very many people to be thanked, but these are just maybe the most representative of the one that came in my mind. I apologize with the others that I wasn't able to remember or to fill in without, but they are all in my heart.